Welcome back, Mushroom Family, to another episode of the Mushroom Revival Podcast. This is a podcast bridging the gap between you, our amazing, beautiful, incredible listeners, and the wonderful, wacky world of mushrooms and fungi. We're unbelievably obsessed with mushrooms and all that they can do and all that they are. We bring on guests and experts from all around the globe to geek out with us and go into the wormhole of all that is fungi. And we're mushrooming the culture around fungi and mushrooms. And we're so excited to have you join us into the mushroom family. So let's tune in and shroom in and get into it.
And today we have the long-awaited topic of the Armillaria genus, also known as the biggest organism on Earth. And we are joined with Myron Smith, a professor in biology who specializes in this genus. And we are going to get deep into the mycology of this fungus and why we love it so much. Myron, thank you for joining us today.
Oh, you're welcome. Nice to be here. Always happy to talk about fungi.
Do you have mushroom magnets on your fridge behind you?
Oh, I have all kinds of things behind me. I have they're not magnets, they're actual real mushrooms. And so I do a mycology class, and so I try this term to add something every day new. And I don't know if the students have noticed, but I try to add some mycological thing every day. And sometimes I use them as props. Like here I have some grass culms with ergot.
Nice.
And, this is ginger. It's actually, the roots have a good antifungal, so I talk about that.
And where are you tuning in from today?
I'm in Ottawa, Ontario, Canada.
Well, why don't we start off with a bit of your story and how you got into fungi? We always ask people how they got inoculated and have decided to devote a chunk of their life to mushrooms.
Yeah, I have really early memories of collecting mushrooms with my family and surrounding families out in the bush in northern Alberta. And, as a child, hunting mushrooms with homemade bow and arrow. So these are some pretty deep sort of memories for me. And then when I started university, I was interested, first of all, in botany because I thought plants would be useful. And then I started getting into mycology because that was where it was taught. And then I was also interested in genetics, and that's where I started my graduate work with Jim Anderson at U of Toronto. He was one of the only fungal geneticists in Canada at the time, so I was pretty excited to meet him and start working with him.
Well, before we funnel into your specific research on Armillaria found in Michigan, I think it's definitely worth visiting this genus as a whole. So the Armillaria is more commonly known as the honey fungus, and it's edible. People forage it. It's found all over North America and I believe parts of Asia and potentially even South Africa.
And Australia.
Great.
Yeah.
Could you give us kind of the 101 on this genus and what sets it apart from other fungi?
Yeah, it's a natural part of the ecosystem. It's probably got a positive role in thinning forests and usually attacking diseased or suppressed trees. But it can be problematic in tree farms and in natural forests. There's a few species of Armillaria that are actually pretty aggressive pathogens, and it does cause damage to trees in that respect. And I think what sets it apart is mainly the rhizomorph structures that are bundles of hyphae. I think it's probably the only genus that has rhizomorphs. There are some woodland fungi that have cords, and they're sort of aggregates of hyphae. And if you dig around the soil in a forest, you'll find, these white strands that are actually bundles of hyphae. But Armillaria has a very advanced type of cord, and it's called the rhizomorph. And that's where it also gets its other name, is the shoestring fungus or bootlace fungus.
Oh, cool. This particular fungus has gotten a lot of notoriety and a lot of spotlight in a lot of news articles that, it's crowned as the biggest organism on the planet. And most people think of that, if they're not mycologists or not too invested into this space, they think of a huge mushroom fruiting body, but it's actually these underground networks, these rhizomorphs that normally people don't think of because obviously they can't see under the soil. And there's a few different colonies. One that I think is the biggest is in Oregon, that I believe is over 2,200 acres. Is that right? Can you just touch upon this specific colony and maybe some fun facts that you can share with our listeners?
Yeah, these are probably organisms that would sort of get to be this size, provided that the forest that they're living in is old and stable and relatively undisturbed. And then they will be able to grow to this size. And it's not really clear why some individuals get really large. By far, most individuals of these fungi are going to be really small. Probably, 99% of the individuals are smaller than, you could hold them in your hand. But every once in a while, for some reason that we don't really understand, some of these individuals get really large. Could just be that they were in there early on when the forest was established. So for example, in northern Michigan, the big fungus we've been working on probably came in after the Ice Age when the trees started to colonize, and my feeling is it got a jump on other individuals, and that's how it got to be that size. So as you say, the challenge is really identifying the individual boundaries. Where you're walking through the woods, you see a mushroom here and a mushroom 10 meters away, and without doing the genetic analysis, you wouldn't really understand that those are parts of the same mycelium, part of the same fungus. So you pretty much have to do the genetic testing, except in the case of the West Coast individuals, you can see those almost like a fairy ring from aerial photographs. But the one we've worked on mainly in northern Michigan is not really obvious from aerial photographs, and you have to get in and do the genetic analysis and a lot of collecting to kind of see where it begins and where it ends.
This is also called the Meadow Maker, and a lot of people are scared of anything parasitic, and obviously it depends on what level of the ecosystem that you're looking at it through, and some people have said, well, that's a good thing, it makes meadows, and a lot of plant species and animal species and bacteria and things like that really thrive in meadow-like environments. Probably not good for the trees, but maybe on a million-year scale, it might be good for the trees. And can you said that you can see kind of like from a satellite image this fairy ring? Is it this destruction of trees, and there's this limit where you can see living trees where the fungus hasn't reached yet?
Yeah, it's a noticeable circle of dead and dying trees that you can make out. You can, when you look specifically for it. It might not be obvious when you're on the ground necessarily, but it's certainly obvious if you look at it from a distance. And other fungi have similar characteristics, like the fairy ring mushroom that you see on lawns and things like that would also grow in rings. So the difference there is that Armillaria is kind of growing out, and it's still occupying the center area, and it becomes maybe quiescent and will attack a tree and maybe attack a sick tree. But they've been doing this in these areas for thousands of years at least. And so, the forests are still healthy in these areas overall. So it's not like there's something unnatural or they're causing a collapse of forest ecosystems. I think that it's more accurate to say that they're part of the natural ecosystem. They're there to convert woody waste material into nutritional elements for other organisms, and they're also part of a rejuvenation process. So, pathogens have a place too. It's just when things get out of whack and out of balance that we really notice them as having a major impact on human affairs. But from an ecosystem perspective, they're a natural part of it.
And I want to revisit the statistic of this individual being the biggest organism on Earth. Correct me if I'm wrong, but that just applies to the acreage that it covers, but if you were to actually bring that individual all together and clump it up into a ball, it would come probably second or third to the blue whale. Or where
No.
would that be?
No, I think
Ginkgo, the aspen, the colony of trees, is the biggest by mass.
But there again, most of the tree is dead wood, so you'd have to really do the arithmetic on that and look at the living wood or the living part of the tree, and then factor that into your calculation. It's not so. We published that paper in '92 where we did the genetic analysis, identified this single individual that had a large area, and then in talking to actually a fellow at the university who asked, well, how big is it? How does it compare to a dinosaur? And how does it compare to the largest animal ever to wander on Earth, the female blue whale? And so, we started looking into, trying to calculate. It's really not easy, and it's pretty rough calculation, but trying to get an idea of what the density of living tissue is in a, 10 centimeter cube of soil, and then sort of multiplying that by the area to get some idea of how massive this individual is. So the mass is in excess of a single blue whale. It's probably more like, so the one in Oregon, I'm not sure. I don't know that the mass estimates have been made, but the one in northern Michigan is in around probably 2 to 4 blue whales in mass. So these are really dispersed in sort of indeterminate growth forms that when you start sort of, as you say, gathering them all together, they would have an incredibly large mass, that's for sure. And they're also really old. Like our estimates of age are based on if there's uninterrupted growth from some center point out to the margin. But that's very naive because we know that there's lots of other individuals around and they are all kind of duking it out for territory. And you can't necessarily push into another individual's territory. There's going to be a battle. There's chemical signaling and toxins being produced and antibiotics. And so it's not easy to, overtake other individuals once those individuals are established. So, our estimates of age in around, 2,000 to 4,000 years are probably very on the low side, I would say.
Yeah, that was astonishing to read that the estimates were between 2,000 and 8,000 years.
Since you're not able to really see it, right? How your paper talked about relying on DNA analysis to figure out whether this is one huge colony versus a lot of smaller ones kind of dispersed about this specific region. Do you know 100%, or is it kind of a best guess, we 99% sure, a high number?
It's almost 100%. You're never 100% in science. You can never, in natural science, you can never, there's no such thing as proof. But so initially what we did is we looked at polymorphic genetic markers. So ones where there's some variability. And then you're right. What we did is we said, well, if it's not an individual, what would be the next, most genetically similar set of individuals? And that would be like progeny or parent offspring type relationships. But when you get sort of that type of relationship, you generally lose genetic variability from individual to individual. So it's the basis for inbreeding depression, for example. So what we did is we looked at these variable genetic markers, and we could use a statistical test to show that in the original paper that over, with a probability of 0.001, that all these samples were from the same genetic individual. But we really locked that down a few years ago when we did the study where we sequenced the entire genome from different areas of the Michigan individual. And there we saw identity almost across this individual. So really remarkable identity, way less genetic variation than I have, say, if we were to sample the cells in my toe compared to the cells in my finger, there'd be way more genetic mutations evident in that analysis than there are across this massive fungus. So that fungus is doing something. We're not really sure what. We can't explain it. We've got some ideas. They need to be tested. But the fungus is not only large and old and massive, but it's also extremely stable from a genetics perspective.
And you brought up that it's kind of rare that they're able to grow this large. And obviously the Armillaria species in Michigan is a different species than the one in Oregon.
That's correct.
But they're the same genus and they're still fairly similar. Do you think the reason the Armillaria species in Oregon, the famous one to be the biggest organism on the planet, do you think that it's more of an aggressive species? Do you think that it's a different tree that it's parasitizing that has less antifungal compounds that it's excreting, or was it already kind of sick, or maybe the trees in Michigan have more antifungal properties, or the fungus is less aggressive, a combination of these factors?
It's all good questions. I'm not really sure, again, why some individuals achieve tremendous size and most individuals will not. And also, probably there are other individuals that are even larger, and because we cannot detect them or, we don't know where they are or identify them, then, we haven't seen them yet. But I think the reason why it captivated sort of the public's attention is that these are really considered microbes. We think of fungi as, we study fungi in microbiology. It's a microbiology lab that I have. But, for microbes, these are really large organisms. And so it sort of then starts to open up questions about, what is an individual if you've got a whole bunch of bacteria cells that are identical genetically? Do they represent a single individual? Does a beehive represent a single individual? So you quickly get into sort of philosophical questions about, our human constructs, about individuality and how do we define an individual, and what is it to be an individual. But I think the difference between this and, say, like a yeast colony, you might consider yeast as being very large individuals that are dispersed across the planet, right? All the breweries are using Saccharomyces cerevisiae, and many of them are from the same strain. So you might think, okay, well, that's a very large individual. It may well be. The difference is that these Armillaria individuals are in discrete areas. We don't know the exact shape, and that shape probably changes from year to year depending upon what trees fall down and what food they have available. But they are in a particular area, and there's probably some communication across the individual. We don't know about that, but that would be something I'd be interested in testing. But there may be some signaling within the individual that allows these individuals to kind of coordinate their activity in some sense.
Is Lidar a technology that's used for scientists looking subsoil at microbiomes and can you use that technology to view the mycelial landscape? I watch a show on Disney Plus that I think his name is Albert Lin. I might be butchering that, but he looks at ancient landscapes like in Colombia and finds ancient cities through Lidar. And it's so interesting to peer through, trees and shrubbery and to see the landscapes non-visible to the human eye. Is it possible to see through soil with Lidar or similar technology to actually see this fungus and in underground highway network that they're forming?
Yeah, that would be really fabulous, wouldn't it? I don't know if it's quite possible. The other thing to keep in mind is that these are not like excluding all other organisms in their territory. There's there's lots of other fungi growing in there, and they all have slightly different, habitat preferences, different niches, and so they're even different species of Armillaria can overlap in space, but they have, maybe one is growing on conifer wood and one is preferring hardwood or something of that sort. So it would be really hard to use a technique like that to sort of disentangle all of that biomass in there and all of the different fungi and be able to identify a particular genotype. That would be fabulous if we could do that. One of the co-authors on our paper, longtime collaborator, Johan Bruin, said that what he would like to do is be able to take a big water hose and just rinse off all of the soil from around, and then you would see all these rhizomorphs, and you could then sort of get an idea of the three-dimensional structure of the organism. But that's totally impractical, and not only that, it would destroy the organism probably. So I think we'll have to wait on that.
It's always a sight in, National Geographic or similar channels when they pour concrete in ant colonies and then they dig it up and you can actually see it.
Yeah.
Is radioactive isotopes a technique to inject the mycelium with some sort of radioactive isotopes and see the colony that way? Yeah,
actually, the other collaborator, Jim Anderson, did that when he was in graduate school, and he showed that rhizomorphs actually, will translocate materials through the rhizomorphs. So using radioactive tracers. So again, though, you have this problem of, in a biological system, in sort of diluting that radioisotope out, and other organisms start to pick it up and cycle it around as well. So you'd need something that stayed within the individual and that you could somehow monitor. And so I don't know of any system like that. I think the one that we use, which is looking at doing a lot of searching through the woods, finding pieces of Armillaria, doing the genetic analysis, is probably the one that's most likely to work. But it is a lot of work. And the benefit, though, is that you can start to, now with high throughput genome sequencing, we can start to look at the way that the fungus grows. So even though I mentioned, there's not many mutations, the ones that we did find in this most recent study show very clearly that the fungus is growing like it would as if you were inoculating it on a Petri plate. There's a radial type of distribution to the few mutations that we did find. So now we can start to kind of track these mutations and get an idea of the movement of the fungus over time, which is really fascinating. But the thing is that these individuals are actually running, in place. The parts of them are proliferating when they find a food source. They're using that food, they're growing, and then when that food source disappears, that area will kind of die off, and they'll send out the rhizomorphs to forage through the soil, and they'll find other food sources. So they're kind of like, constantly rejuvenating and dying off within their area. So it is remarkable that they managed to maintain these territories, given that, you'd think somebody else would be able to get in there and start nudging them out of the way, but they seem to be, they have some tricks up their sleeves somehow to keep that territory clear of other individuals of the same species.
Yeah, and one of the things that makes Armillaria so powerful are these rhizomorphs. Yeah. And you mentioned putting them on agar. This is actually my favorite fungus to view on a Petri plate. It's looks like these thick black tentacles. It hardly even looks fungal. It just looks like a baby squid or an alien. Yeah. It was very satisfying. I didn't know that the Armillaria would form those rhizomorphs on agar. So
yes.
Is it ever filamentous? I mean, initially when the spores germinate, I do see that. But within the soil, is it usually always rhizomorphs?
Yeah, the rhizomorphs are probably primarily a foraging structure. So they're very good at moving through soil. So they're good at moving from food source A to B, through soil that might be maybe dry or have very little food. So they can move through that desert land and find, another wood source and feed on that. But as soon as they hit the wood, it's really quite interesting. They behave like people would, like a kid at they give a bag of candy or something. When they hit a piece of wood that is their preferred food, the rhizomorphs will curl around it. They'll grow and they'll branch and they'll basically surround the piece of wood. And then once they've got it all surrounded and the whole area sort of contained within this rhizomorph network, they will de-differentiate and start sending in individual hyphae to penetrate into the wood and start decomposing the wood. And then they will move those nutrients around and they will build up enough food mass that they can then build more rhizomorphs and then forage out further and find other trees. So the rhizomorphs are largely a foraging structure, but they're also probably a perinating structure. In other words, they can withstand adverse environmental conditions like probably drought and poor food conditions, maybe fires. Like we know in this forest in northern Michigan that we've worked in, there's been several forest fires that have come through there. You can see charred stumps and things like that, but these rhizomorphs being underground are quite sheltered from the heat of forest fires. So those are, and also the rhizomorphs are probably pretty good at resisting other microbes that might be considered disease organisms or something of that sort. Yeah, but they're fascinating structures, and you can grow them in the medium. Some species form rhizomorphs more readily than others, some Armillaria species. Some of them just love to make rhizomorphs, and they make great pets.
Are all of the rhizomorphs melanized?
Yes. Yeah. So just the outer rind is melanized, and it's a kind of probably a defense against other organisms that are trying to gain entry. Melanin is a pretty good defense against UV radiation, but being under the ground, that's probably not such a big problem. But it's also good at protecting the organism from reactive oxygen species, and it's kind of hard and it's like a bit like charcoal. It lasts for a long time. And then inside that rind of the rhizomorphs are the white hyphae. And if you want to get a culture of Armillaria, what you can do is take some of the rhizomorph that you find in the woods, really easy to find, and clip it into like a 2 centimeter piece and put it into a dilute bleach solution for maybe 5 minutes, pull it out and just blot it on a Kleenex, which is actually pretty sterile, and then clip off the ends and put it onto the medium. And what you'll see in about 2 or 3 days is these kind of a dumbbell shape, but where the white hyphae emerge from the ends. So the rhizomorphs, where the melanized tissue is, they don't really grow out from there. It's almost like a barrier for them to grow. But where they do grow is right from the tips, from the two ends. So you get this, this black rhizomorph with two rapidly growing puffs of white mycelium coming out of that. So they're really very special structures. And, the other beautiful thing about them is that you can go out at any time of year, except maybe when there's a lot of snow, but even then, and find rhizomorphs, and you can study them. Whereas a lot of fungi, you have to wait for them to make fruit bodies to, find them.
That's a great point. They're very unassuming. The first time I saw these rhizomorphs, it just looked like twigs.
Or roots. A lot of times when we're hiking with people that are, don't like mushrooms as much as we do, or fungi, we point them out and they're so confused that that's actually mycelium, and it's really unassuming, or it looks kind of like a little vine creeping up a tree or something like that. Mm-hmm. Are they the only genus that makes these melanized kind of sheaths or protective barriers in their mycelium?
Yeah, as far as I know, like I was mentioning, there are some woodland fungi that make cords, but they're nowhere near as complex as the Armillaria rhizomorphs with the melanized outer rind, and there's at least 2 different cell layers in there, and the tips are a little bit like meristems of plants. They have special features on the tips, so they are really specialized structures for sure. And what I usually see, the ones in the forest, may be 1 to 2 millimeters in diameter, and some are much smaller, even 0.5 millimeters. So that's pretty spectacular. I've never seen a rhizomorph quite like that. That's for sure.
So you already kind of alluded to what we could expect to see if we were to have a cross section of this and look inside of the rhizomorph, and it would be a bunch of hyphae. But would this be like conduits of each individual hyphae, or is it more knotted up, or are the hyphae more of a rhizomorph within a rhizomorph? How does it look?
Yeah, so if you were to, for example, roll over a log and find a rhizomorph, some species have a kind of flattened rhizomorph and some are quite cylindrical. So if you took a cylindrical one and you were to look at the individual hyphae in there, there'd be bundles of hyphae that are arranged sort of parallel to one another, of various different lengths, the individual cells, but overall the hyphae are all running parallel and quite tightly bound together. So there's probably some sort of adhesiveness to the system, and then they're all wrapped in that melanized sheath.
And what exactly is the melanized sheath? Is this just a matrix of a pigment? Yes. With some chitin?
Yeah, so melanin is really interesting. A lot of fungi produce melanin. There's a whole bunch of molds that produce melanin, and it's recently been found that probably these melanized fungi are actually able to use energy from radionuclides, like radioactive energy, to fix inorganic carbon, similar to how plants use light to fix inorganic carbon. So it's has many different functions. It's something that we see when 2 different fungi meet each other. They often will put down a melanized zone. It's often in the spore walls to protect the spores, it's thought, from UV radiation and maybe desiccation and other things as well. So the melanin has many purposes, but it seems to be really useful to the fungi, and it's rather specifically controlled. There's only a few places where Armillaria lays that melanin down, and that's in the outer sheath, and also these things called plates. When 2 individuals meet each other in a log, they'll set out like a kind of if you could look at it in 3 dimension, it would look like a sheet of melanized tissue that's separating the 2 different individuals in the log.
So this is between 2 different Armillaria species?
Well, 2 different species or 2 different individuals. So 2 different genotypes, and they will lay down a barrier. It says, this is my turf, that's yours, and stay over there.
It's interesting because the whole melanized sheath, it makes me think of a wire for electricity, and kind of this rubber
yes,
outer coating of a wire. I actually just searched on Google, so I haven't researched it too much, but apparently melanin, or actually a form of melanin, is can conduct electricity. And I looked at one paper just really quick that a positive electrical charge from a 9-volt battery inhibited the growth of Armillaria meliae colonies by killing the tips of the rhizomorphs. So I've seen a lot of research recently on electrical conductivity of fungi for replacing parts of batteries.
Oh yeah.
And have you heard of this specifically with Armillaria and kind of maybe in nature, but also use in biotechnology?
Not so much applied, but there was a study probably about 1989, 1990 by Olsen, I think was his name, from maybe Denmark. And he showed that there's an action potential in the Armillaria rhizomorph. So an action potential is a normal sort of charge-discharge process that many living cells will do. And I really like this study. He basically hooked a extracellular electrode up to a rhizomorph, and he started getting the signal, which is like a tick, 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 tick signal as the depolarization occurs in the cell. And then when he put a small piece of wood on the rhizomorph, it started going tick, 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 tick. And when he put a little bit of sugar, and this was about a centimeter away, there was a very brief pause, and then it went.
Wow.
So his study was published in Naturwissenschaft, and you might have a look at it. It's really under, it's kind of gone under the general notice, but it does suggest that there may be some signaling that goes on, maybe electrical signaling through rhizomorphs. And rhizomorphs also will branch. You were asking about the shape of them. So they will branch, and they can take on really lovely sort of antler-like shape. Some of them are bifurcating, some of them are more like a root sort of branching system. So I had an artist in residence here that worked with me on Armillaria, and she produced some fabulous artwork based on the rhizomorph structures. So, there's a lot of maybe application in that area as well.
So if mycelium is the wood wide web, then the Armillaria rhizomorphs are the Ethernet.
Yes. Fiber optic system, yeah.
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 Armillaria is so impressive. It's the biggest organism, one of the oldest living fungi, contains this very powerful protective melanin sheath, and it also glows, correct? Do all Armillaria species glow? And is this just the gills, or where exactly is it?
Yeah, it's bioluminescent. So that's a process that actually requires oxygen. It requires, a living organism, and it's not fluorescence, it's actual bioluminescence. Just like a fruit fly, a firefly. Yeah, firefly. Glowworms, many bioluminescent microbes in the oceans and stuff like that. And Armillaria, I think there's only one species that I've seen of Armillaria that I was unable to see bioluminescence in, but all the ones in North America that I've looked at are bioluminescent. And you need you just have to grow them. They really like it if you grow them on breadcrumb agar. So if you take some whole wheat bread, let it dry out, put it in the blender, make some crumbs, and then add maybe a little bit of sugar and some agar and pour some plates out of that. You can inoculate Armillaria on that. And once they get to be, about the size of a quarter or so in colony diameter, and you go into a dark room, give your eyes a few minutes to dark adapt, and you can see this kind of eerie green yellow light being produced. And it's so bright that I used to actually take notes in the dark room with a stack of plates beside me. I could write quite easily once your eyes are dark adapted.
What?
Yeah, it's pretty
fabulous.
It's
the mycelium as well. It's not just the mushroom.
So the mushrooms, as far as I know, do not bioluminesce. So there's another group, the Pinellus, that does. The fruit bodies do bioluminesce. There's probably only a handful of genera of fungi that I know of that bioluminesce appreciably. And Armillaria certainly is one of them. So it's really the mycelium, and it's in a particular area. It's usually in the growing front of the mycelium, where there's lots of white hyphae being produced. Those will be spectacularly bioluminescent. So when you look on the plate, often you'll see a kind of a ring of light being produced around the periphery of the colony. Not really clear what the function is. It could be partly to attract insects. It could be aposematic, in other words, a warning signal for some organisms. It could be simply a way of drawing unfavorable reactions into a downstream sort of process. So a bit like generating heat will pull reactions that otherwise wouldn't normally occur. But it is another mystery about Armillaria, why it bioluminesces, and it's a pretty cool attribute for sure.
I've always wanted to take a bunch of Petri plates and then tape them to my ceiling in my room, like those glow in the dark stars and moons and stuff. If only we can get star and moon shaped agar dishes. I mean, I guess you can get the full moon, you know.
Yeah.
You could do like a tessellation to make it. It'd be very large. But
yeah. I'm curious as well, because one of my mentors and teachers for the last few years, Trad Cotter, he, in one of the classes I took many years ago, he said, you know, for a lot of fungi, think of the fruiting body as almost like an escape pod. And a lot of times the fungi will create fruiting bodies almost as a defense mechanism, as I'm hitting another fungi, or some competition or a barrier of a rock face, or I'm running out of food. I've already colonized this log and I know that I'm running out of food, almost like the biological mechanism in humans of I've reached a certain age, I know I'm about to die, or, you know, some dinosaur or some wild creature is going to kill me any day now, or I'm going to starve to death or whatever. I must, you know, make sure my DNA gets passed on. It's a biological just phenomenon. But for, you know, the largest organism on the planet, if they, this goes into the kind of like the psychology of fungi, but, you know, if they have an abundance of food, and I'm just curious about the amount of fruiting bodies in that colony versus another one that maybe is smaller, that maybe is facing more competition, who's maybe has less food available. Do you see more fruiting bodies in the smaller colonies as you do the big one?
Oh yeah, you can get Armillaria to fruit on an orange, for example. They quite like oranges, and they'll fruit on, you know, you autoclave an orange and you inoculate it and grow it at about 23 to, you know, room temperature for until it's colonized, and then switch it down to about 17 and put a day-night light on it, and you can get fruit bodies forming, especially some of the species. And it's true with
We're doing that.
It's true with most fungi and maybe most organisms that as soon as they get stressed, they go into a reproductive mode. So, you know, end of the season, they're running out of food, conditions are becoming adverse, maybe there's competitors or something of that sort. They'll go into a reproductive mode. And it seems that some fungi are, you know, sort of adapted to colonizing really small food sources and then and growing very quickly and then sporulating very quickly, and kind of like a, you know, a strategy there. But other ones are more, you know, long-lived, and they're going to grow a lot longer. And so it seems that Armillaria has almost got both of those worlds worked on. These things produce tremendous numbers of spores across that individual. Can you imagine? Like some years we collected hundreds of fruit bodies within a few square meters. And so if you kind of then, you know, multiply that by the entire area, then you have an awful lot of spores being formed by that individual. By and large, probably almost all of those spores will be unsuccessful. You know, they won't find a suitable habitat. They'll be lost in a stream or a lake or something of that sort. But you know, they are definitely of the mindset, if you will, that they're going to produce a lot of spores, and maybe some small, minuscule proportion of those spores will actually be successful. And it usually is when there's a stressful condition that they start fruiting. And you can grow Armillaria for years in the lab without it ever fruiting. You know, it's fine. You just keep transferring it from feature plate to feature plate, and you can put it in the fridge and keep it for a while. And, you know, so it's not like they have to fruit, and they do have to have a certain amount of biomass, it seems, in order to make a single mushroom. And the mushrooms that I've gotten in culture are nowhere near as robust as the ones that we find in the field. Like, they're not as dense. They're kind of watery. You know, they dehydrate almost down to nothing compared to the ones in the field. So There's probably a lot of nutrients being pumped into those fruit bodies in the fall when they're making them. They're investing a lot of energy into them. They're not getting a lot of return, but you're right. It's the only way they're going to move forward if they want to colonize new space, or if somebody comes and mows down the forest.
Right. So I have kind of a heady mycology question for you. And in your paper, you described the way that the Armillaria reproduces, and it is a basidiomycota. So most basidiomycetes, two compatible hyphae, will come together and go from monokaryotic to dikaryotic. So they had one nucleus, and now they have two per filament. But Armillaria doesn't do it that way. They seem to maintain their haploid mating or haploid state, and then don't fuse until they want to fruit. Or can you explain this behavior? It almost sounded like ascomycota to me when I read about it.
Yeah, so it seems that, again, Armillaria is a little unusual compared to other basidiomycetes in that the most common vegetative state is a diploid instead of a pair of haploid nuclei, instead of a dikaryon. So the life cycle of Armillaria then is that the basidiospores are produced by the mushroom. They're on the underside in the gills, and they kind of are launched out of that area. They fall down beneath the mushroom, and then they're taken by the wind. And when two basidiospores germinate in close proximity, chances are they're going to be compatible mating type. There's hundreds, if not thousands, of different mating types. It's not like they have a male and a female sex. They have thousands of different sexes. They're not, there's no problem with finding a partner for these guys. So the two germlings from the basidiospores will grow together, and they'll fuse, and then they will probably form a brief dikaryon, but then the two different nuclei fuse to get a diploid, and that will grow as a vegetative mycelium that's capable of fruiting to form mushrooms in the fall. And so what happens is the diploid nucleus will go through a regular meiosis, form four different nuclear types that are partitioned into four basidiospores, and then they will be airborne again. So the real unusual part of Armillaria is this tendency, it seems, to maintain the vegetative phase in a diploid state. So there is some controversy over that, and I'm not really sure if the jury's out, but when we look at it in the lab, at least, it is a diploid. It maintains a diploid. In the field, there has been some dikaryotic hyphae detected. So it seems like, maybe it doesn't have the same issues with, having those nuclei stay apart and maintaining a dikaryon that other basidiomycetes do. So it is another attribute. You're starting to point out all these weird things, and now I'm starting to think it is actually pretty weird even for a mushroom, even for a fungus. Yeah.
Yeah, it's a weird species, a weird genus, but it's one of my favorites because of that. It has so many unique characteristics.
Yeah, it's true. Yeah.
I went to your website and I looked at all the other research that you do in the breadth of how immense fungi is, and I'm curious because I found like over a dozen papers on antifungal properties of plants. And I'm just curious if you got into that through Armillaria, just researching this massive organism that's just plowing through trees and is thousands of years old and thousands of acres wide, and just that point of interaction between plant and fungi, and that chemical battle that takes place. Did that spark your curiosity of what compounds are plants even using to combat fungi?
Yeah, that's part of it. The general area is natural products, and the idea is to be able to use natural products to influence our surroundings, to fight off microbes and that sort of thing. So it's true that was something that I was a little bit mystified when we were studying this huge individual in Michigan. We started to, understand how large and how old it is. And then you start to think, well, what are the attributes of that individual or species that allow it to get to that size? Most organisms, get sick and die, right? But these individuals are able to grow, seems like almost immortal, and to be really stable over long periods of time. So they must have some pretty interesting chemical defenses. And the other part of that sort of sparked my interest going forward is that they must have an incredible sense of self, or an ability to distinguish between non-self. So if you think about our genetic system, we sort of constantly being bombarded by viral elements and transposons and all these little pieces of DNA that insert themselves into our genome, sometimes inserting into genes that cause diseases and genetic diseases and cancers and things like that. And we cannot necessarily identify, for example, a cancer cell from our own cell. Our body doesn't have the discrimination to be able to tell a healthy cell from a non-healthy cell in many cases. But it seems that these large individuals can do that. So that, to me, sparked my interest in non-self recognition, which is a kind of immune system that fungi have. That fungi are close relatives of animals, much closer than they are to plants. And so we should start looking at what we know about animals to understand about fungi more. And we should start looking at fungi to understand more about animals, because they're sister taxa. And so when we know a lot about the human immune system, we can probably apply some of those lessons to understand the fungal immune system. But it's clear that it's highly developed, highly evolved, and really good at its job. So getting back to your question about antifungals, the other thing that sparked my interest is actually a book I got when I was 11 years old. After a visit to the dentist, my parents got me a book, and that book was about ethnobotany, and I always carried that with me. And so when I had an opportunity to study medicinal plants and medicinal fungi, I took that opportunity as a professor. So that's funny that you look at it.
Do you remember what the book was?
I sure do. I have it right here on my bookshelf. Yeah.
What book is it? I'm just curious.
It's called, I think it's called American Indian Medicine. And it was based on a Ph.D. thesis. And so it was pretty thick reading for an 11-year-old. But there was some pretty cool parts in there that I enjoyed. So, a lot of times when you're kind of moving through your learning experience, something opens a door and you discover an incredible world in there. And, it's a fascinating and really a privilege to be able to walk into those doors and discover new territory as a professor and a scientist.
What doors do you want to see opened?
Oh, lots.
Maybe, and for Armillaria, but then also just the kingdom of fungi. Yeah. Of, what research are you dying to see done?
Yeah, well, there's, I think the big unknown right now is really fungal ecology. And we have now some tools available that we never had when I was coming up as an undergraduate going into graduate school. At that time, there was not even a way to identify how many individuals of a particular species you had in the forest. So how can you do any kind of, analysis? It's really problematic. Now we're getting to the point where you can get a genome sequence overnight almost. And so, and we're getting a lot of analysis pipelines in place. So, we can use those tools to start to look at, classical questions on ecology with fungi. And I try to encourage my students to do that, because I think there's some really valuable lessons for humanity in the way that fungi have survived and persisted, and they're so diverse and they do so many interesting things, and I think that we can really ally ourselves with fungi and build interesting systems and products and foods and packaging, and just with it's a really undervalued resource that I think we can access. Yeah, so the doors that I'd like to see open, there's many. That's one. I'd like to see more studies on ecology of fungi and how they interact with other organisms. I'd like to see more on communication within individuals. This idea we're talking about, whether there's signaling across an individual, whether, we would joke when we go out into the woods. We initially did our work we started in 1989, and we finished that first study in 92. Oh, it might have been 88 or so in 92. And then we took a break until, almost 30 years later, and we went back to the site. And when we got out of the vehicle, we were kind of going, oh, gee, I wonder if it remembers us, I wonder, like, you don't know about, because we're dealing with an organism that's on a totally different time scale. It's really not very similar in some ways to animals, which we understand because we're animal. And yet we know that they're a sister taxon, so probably there are some similarities. And there's some pretty cool examples in the mycological world of, fungi having the ability to sense chemicals, so they have a sense of smell or taste. They can perceive structures, so they can touch. Like rust fungi know when they're going perpendicular to the host cell versus in the same direction, and they have light receptors, and some of them can aim their spores and shoot towards light. And so all of these little parts make you think that they're quite sophisticated. And I would like to see some studies testing whether there's long-range signaling. We know that transport is occurring over long range of metabolites and things like that. But is there something faster? Is there more of a fast response? I would like to see some additional work on biotechnology and fungi. I think there's tremendous research. We've seen ideas about packaging and building materials with fungi, but I think there's an awful lot of innovation that can be done in that area as well. So yeah, when I go through this mycology course that I do, pretty much every lecture I say, wouldn't it be great to be able to understand something more about this phenomenon in fungi? So just alone the knowledge generated by studying fungi is spectacular. But there's a lot of uses. We have to be able to figure out how to defend ourselves against food spoilage and plant diseases. And, we need a lot of people working on these areas because those could have a pretty profound impact on our future.
Mm-hmm. Well, thank you for spreading the word and probably inoculating a variety of students to jump on this mycotechnology revolution.
Right. Yeah.
Yeah. Is there any last thing you want to share with our listeners about Armillaria?
Well, it's one of many really interesting fungi, and there's you could probably throw a dart at the fungal tree of life and you would come up with some really interesting stories. I'd say read some of the old literature. Some of the there's a professor in Winnipeg named Buller that published a series of books in the 1930s, around there, and you just crack open one of those books and he made some observations that are pretty amazing, and we now have some tools that we could start to look at those questions. So and I'd say do some exploration, have some fun, and use your imagination and intuition to follow what interests you. And I think that it's a pretty spectacular field to be in right now.
Oh yeah.
We always ask a final question that opens that door to create an imaginary space. If we could communicate with fungi and we did have the sensory receptors and tools to be able to connect with them on a deeper level, it's always fun to ask this question, but do you want to take it?
Sure. The question is, if mushrooms had the microphone or fungi had the microphone and could say one thing to the whole human race, what do you think they would say?
Well, they might say, use your resources wisely. Armillaria is really good at pacing itself, and it's in there for the long run. And I think that there's a lesson in that. It probably has a lot of advice. If we just kind of let that mic stay open for a while, it would probably have a lot of advice to us. Another one would be, don't get hung up on human constructs.
Those are great. I love to preserve your resources. We're eventually going to compile everyone's answers and maybe publish a little zine or just an infographic of responses.
Hopefully one day we'll figure out how to plug in the Armillaria Ethernet cord into our computer, and
that'd be great.
Yeah, into our Neuralink. Just plug it into our head.
Yeah, the one in the lab will probably say, "Let me free."
Right.
Definitely.
Where can people follow your work?
It's pretty outdated. I was actually today I was planning on updating it. I've noticed I haven't updated it since 2013, so that's kind of embarrassing. I guess that, or they can contact me through Carleton University in Ottawa. There's also a Carleton College in the U.S., but this is the university in Canada, so they could do that. I don't know. I'm pretty keen on fungi as well, and and I think that how long have we been talking here? About an hour?
Yep.
About an hour.
Yeah, so it's gone by really fast, eh?
Mm-hmm.
Just because you start talking about these things, and you could spend a lot of time on it. So it's great that you're doing this program.
Yeah, people are really loving it and learning a lot, and I think it's inspiring a ton of young people to get into this business academically in the startup world.
Yeah, there's all kinds of career paths out of mycology. Yeah.
And likewise, we need more fungal educators.
Yeah.
I hear it all the time of people wanting a course or wanting some sort of deep dive into fungi, and there's not much out there.
No.
There's some books, a lot of research papers are under lock and key or a hefty price tag, and
right.
Yeah, and so to be able to spread this passion and this magnifying glass on this whole just untapped, it's huge queendom that it's just, we don't know anything about it, and we're just scratching the surface. And like you said, you just throw a dartboard and the first thing that comes up is probably the coolest thing on the planet. And it's fun to be in that frontier and to be those explorers into a field that is pretty untapped, and anywhere you go into it, you could be the first in the world with your research, and really help develop this field from the ground up, and our human understanding of what the world is around us.
That's right.
With that said, thank you everyone for tuning in and trimming in. That wraps up another episode of our podcast. If you are listening to this on, say, Apple Podcasts or Spotify, there is a video component. So head over to our website at mushroomrevival.com or YouTube. You can see the video there. And if you're already watching, hello, thanks for tuning in. We'd love to hear your feedback. So if you want to reach out to us or leave a review, leave a comment, tell us future topics that you want us to geek out about, or guests that you want us to bring on our show, or just to say hi. We love mushrooms. We love the mushroom family, and we want it to keep growing. So tell your friends, tell your family about how cool fungi and mushrooms are. And let's keep researching. Let's keep exploring together. So as always, much love, and may the spores be with you.